Hello again, this is Mark Armitage coming to you from the microscopy facility in the biology department at California State University, Northridge. We wish to acknowledge the Keck Foundation and their financial support for the production of these videos. We also want to acknowledge Nikon Microscopy University for the images which we're going to show you today. We will begin again with an overview of the confocal microscope, the Leica SP5, and then we will delve into more specifics about individual components of the microscope. To initialize the confocal microscope, you must first apply power to the microscope and to the computer which drives the software. You also would apply power to the lasers, but we will hold off on that until another segment. The Leica Confocal Microscope has at its base an inverted microscope. Now previously we discussed an upright compound microscope. And so we discussed the illumination path, we discussed the optical path, and here with an inverted microscope, everything is reversed. Everything is upside down from the upright microscope that we worked with previously. For example, the optics are below the stage. So all the objectives that you use for imaging are below the mechanical stage. The condenser is above the stage. Before, the condenser was below the stage and the optics were above. These are reversed now. The transmitted light illuminator is now above the stage. And so instead of transmitting light through the base for bright field and other forms of transmitted light microscopy, we transmit light from above and down through the stage into the optical system and into the body of the microscope. We discussed epi-illumination, which is light from above for the fluorescence microscopy application. Here we have an illuminator which is supplying the fluorescent light, and again it comes in below the stage. It is epi in that it does come through the optics and reflects off the specimen and back down through the optics and then transmitted into the microscope. But it is reversed because this is an inverted microscope. The transmitted light illuminator has a lamp house back here, and this is where the halogen 100-watt bulb is. When you turn on the microscope, this bulb is on all the time. And so in a few minutes I will show you the intensity control switch to turn down this bulb so that it's not burning all the time. The light comes out of the lamp housing and it goes into a collector system. There are two flip switches here, but those you don't need to play around with. We'll discuss those later. The light comes through this device and comes down into the condenser. Now the condenser does what we talked about previously. It condenses the light and so that it goes into and through your specimen on the stage and into the optics. It is very, very important to align this condenser properly. And we will be showing you two alignment tools that go into these little slots to align the condenser in this plane. In addition, there's a knob on the other side of the condenser, which you'll see in a second, which allows you to raise and lower the condenser for correct positioning of the condenser. This is a knob then that allows us to raise and lower the condenser so that when we align our field diaphragm, it's nice and sharp in the field of view. And so the field diaphragm on this microscope is manual and it's located here. And so we would close this down and open it up so that we could see it in our field of view. And so we're going to discuss condenser alignment specifically as we did on the upright compound microscope, aligning the condenser, closing the field diaphragm, and then using the alignment tools in here to align the field diaphragm to the field of view. Another important feature of the Leica SP5, particularly related to inverted microscopes, is that the illumination arm can rock out of the way. And this is important because often we work with large culture dishes that fit in here on the stage. We have these two inserts that allow us to put our slide in here with our specimen on it. But if we're working with culture dishes, if we're growing cells in culture, then we will need to open this up so that we can put a multi-well culture dish in here. Additionally, this whole insert pops out, and there are other inserts available that fit into the stage and allow us to use different size culture dishes, whether square, rectangular, or circular. And so with an inverted microscope, we have the ability with stage inserts to use multi-well dishes within which we grow cells. So if you're culturing cells, you can use a small dish that will fit into this space, or the entire insert will come out and you can use larger dishes, or we can purchase additional inserts to accommodate the kind of specimens that you're working with. The other feature of this insert is that it allows you to close these sliding holders down so that you can put your slide in there for observation of specimens on glass slides. The mechanical stage on the Leica microscope is motorized. There are motors that drive this stage in X and Y. And so that's another important reason why the condenser has to be rocked out of the way, because when you initialize the microscope, the computer will drive the stage in X and Y to its endpoint because of the motors that are on each axis of the stage. So be aware that this is a robotically controlled motorized stage which can injure your fingers. You could actually lose a finger if you put your hands in here while the stage is moving. So when the stage is moving, keep your hands and everything free and clear from the stage. Below the stage is the nose piece, which holds all of the objectives. And you can see that there are quite a few objectives on this nose piece. This is a motorized nose piece, but we will be manipulating it manually to choose the different objectives that we work with. Later on in this sequence, we will discuss every objective on here, the magnification of it, and the particular parameters of each of the objectives. Several of them are immersion objectives. They might be water immersion, glycerin immersion, or oil immersion, and we will discuss all these at length. It is very important to keep in mind that before you initialize the mechanical stage, you dial the objectives out of the path. It's usually best to dial it to this blank spot. That way all the objectives are below the stage and protected when you initialize the stage and the motorized function takes over. Pay close attention to the nose piece as I'm going to actuate the focus drive on the salt and pepper shaker, and you can see as I manipulate the focus drive, the nose piece goes up and down. This is very important to understand because you can very easily and quickly drive the nose piece into your specimen that's held in place above the nose piece. So take particular care when you're using the salt and pepper shaker to get the right focus. Please understand that the table that supports the microscope is an air table. It's designed to reduce any vibrations that come from the building or traffic outside. If you press on the table, it will decompress and come back up. But we don't want people leaning on the table because that over exercises the air pump that keeps the air under the table in order to reduce vibrations. This LCD screen on the front of the microscope gives you specific information about different parameters of the microscope use. You'll see immediately the flashing letters say transmitted light bright field, and so the microscope is in transmitted light bright field mode, but it's flashing because we don't have an objective in the beam path yet. But it describes the information about the objective, whether or not the magnification changer is engaged, the intensity of the illumination, the size of the aperture diaphragm, and so forth. And so you will consult this screen from time to time while you're working with the microscope to make sure that all the parameters are correctly set. When the microscope is first initialized, it is important to look at this very bottom line of the text on the LCD screen. This line describes the Z position, or the vertical travel on the nose piece. You should be able to see two distinct lines, straight lines, one between these two arrows and one below this arrow. This means, of course, that the Z-axis has been properly set. If you do not see those two straight lines, go get the confocal microscopy technician and have him correct this parameter. If you are working with slides, You must flip your slide upside down because it is an inverted microscope. And so therefore, the part of the slide with the cover slip faces down onto the optic system. Make sure that you put your slide in that configuration or you will not see your image. Then you are free to dial the nose piece and bring the first objective, a 10x objective, into view. The Leica Confocal Microscope has a series of 5 objectives. The lowest power is a 10x bright field objective, and it's a dry objective. The second objective is a 20x dry objective. The third objective is a 20x, but this is a wet objective. Therefore, it's an immersion objective. And so you would put a drop of oil on the front element of the lens before you put your slide down onto the objective. The fourth objective is another oil immersion objective. This is a 63x oil. And finally, the fifth objective is a 100x oil objective, also immersion. Because the 63x objective and the 100x objective are very expensive objectives, they have a unique feature that allows you to lock the front element down and out of the way so that when you're rotating the nose piece, these objectives will not come in contact with the slide. The 63x objective does that, and also the 100x objective locks down and out of the way. Please keep that in mind, as a previous user may have locked it down and out of the way, so that in order for you to do your work, you have to unlock the objective and let it come back to its full height in order for it to work properly. The Leica SP5 inverted microscope is a fluorescent microscope, and therefore it has its own mercury lamp house with burner. To turn it on, you actuate the power button. Notice right away that the shutter button blinked while it powered up. However, the shutter is not open because the light is not on. If you want light to leave the lamp house, you have to press the shutter button. Then the shutter button light illuminates, indicating that light is now leaving the transformer. This is an HBO mercury bulb, such as we discussed before. However, this one's the new variety, and therefore you can change the intensity of the illumination, and you can also turn it on and off. However, it's best to just turn the power on and leave the lamp house running the whole time that you'll be doing your experiment. The shutter lamp is a very important feature of this illuminator. You notice now that the light is off, indicating that the shutter is not open. To open the shutter, you press the shutter open button, and now the lamp is illuminated, indicating the light is leaving the transformer and going through the optical waveguide, which leaves the back of the transformer and travels into the epi-illuminator of the microscope. Now, this waveguide contains an optical gel which transmits the light efficiently. However, if you leave the shutter open all the time, this will degrade the gel inside the waveguide. The gel is very expensive and difficult to replace, so if you're not actually using the fluorescent illuminator, actuate the shutter button and turn off or close the shutter so that no light is escaping the lamp house. Now that our illuminator for epifluorescence is turned on, we're directing light through the waveguide and into the epi-illuminator of the microscope. The light comes down through the body of the microscope, and it actually travels through a filter pack that's built into the body of the microscope. We have 4 filter packs in this microscope. The light travels through the filter pack and into the body of the microscope, and we can send that light to the specimen by pushing on the shutter button. And here you can now see that the light is illuminated on our specimen. We have 4 illumination methods for fluorescence microscopy on the Leica Confocal Microscope. The first one, as you see here, is for FITC. We also have filter packs for rhodamine, for DAPI, and for green fluorescent protein. To actuate the filter packs inside the microscope, you press the buttons on the front of the microscope, which allow you to select one of the 4 filter packages. This button is for FITC, this button is for rhodamine, This button is for DAPI, and this button is for green fluorescent protein. Do not touch these two buttons on the front of the microscope, and only rarely will you actuate one of these buttons, which we will discuss at a later date. 4 important buttons are found on the left side of the instrument, just inside the focus knob. The third button down allows you to actuate and toggle between the different filter cubes, just as you did on the front of the microscope. This is very convenient. If you're using 2 different fluorescent filter packages, you can toggle between them simply by pressing the third button. The first button allows you to toggle between the different transmitted light illumination methods that you're using. For example, bright field, DIC, or polarized light. The second button down allows you to go from transmitted light to fluorescence. And so you can toggle between your transmitted light method that you've selected and the fluorescent method that you've selected. So for example, if you're going from FITC back to bright field or DIC, with one simple button, you can toggle between those two. Finally, the bottom button is very important because the bottom button allows you to switch between observation in the microscope and sending the image over to the computer. So when you push the bottom button, it sends the image to the computer for scan mode. You can also go from scan mode back to direct observation through the microscope by pushing that bottom button. On the right side of the instrument, just outside the focus knob, are 3 important buttons that have to do with setting the Z-axis up and down position. Please do not touch these buttons, as this has already been set by the technician. As indicated, by pushing the top button, you're toggling between the different illumination methods, transmitted light illumination in the microscope. And you can see this on the screen. The top line shows that I'm in transmitted light bright field. If I push the button, it switches to DIC. If I push the button, it switches to polarized light. And so by actuating the button and looking at the top screen, you can see what transmitted light illumination method you're using in the microscope. When I rotate a particular objective into view, I can see on the screen the objective that the microscope is detecting. So for example, now it's the 10x objective. If I rotate again, it shows the 20x objective. The screen also shows you if the 1x magnification changer is in place or if it's set to scan mode. Often when it's set to scan mode, the microscope, when you come back to visual observation, you must actuate the 1x magnification changer so that you can see the image in the field of view. We'll discuss that more later.